All right, so as you can see, we put a few of these vocab terms up here, and I'm sorry, I erased a couple of them. I'll put them back up here before the class is over, okay? So if you're having problems reading this, you might want to hit pause. It just says a nucleic acid that is similar to DNA, okay? Transfer RNA are smaller segments of RNA nucleotides that transfer amino acids to the ribosome. Codon, 3-base code in DNA or RNA. Translation, a process in which mRNA attaches to the ribosome and a protein is assembled. Okay. Transcription, a process in which messenger RNA is synthesized from the template of DNAs, is what I would think would probably make more sense. Okay. So, next, open up to page 336, okay? So what process have we put up on the board a couple of times now? No. Yeah, what is A, T, C, and G? What process did we illustrate on the board last week? Starts the letter R. The example we gave was if you have a serve and you get an ace, you want to do that again. So you replicate it. You replicate it. And you replicate it. So what does that mean? What process was that? It was what type of replication? 3 letters long. Just back up a couple pages and it'll tell you that. Remember, it's got the alligator jaw opening up, or the replication fork. At least I think it would be a page prior. Yeah. Says it here in blue. DNA what? DNA replication. That's right. Okay. Now, on page 336, okay, DNA, RNA, and protein. One of the things that 336, okay, one of the things that I don't know as far as proteins are concerned, I like to talk about venoms quite a bit. Does anyone venom? When venom is just kind of a vague term, where do you find a substance like venom? Snakes could be one. Lizards, fish, even mammals. There's one species of mammal, okay, that actually has venom in it, okay? Now, some of these venoms can be used for treatments and medical purposes, but can anyone, not that you're going to know the answer, but why don't we replicate these venoms? And the answer is they're just way too complex, okay? Venoms are proteins. How do you get proteins? Well, that's what this process deals with. It's called protein synthesis. That's why it says DNA, RNA, and protein. So if you synthesize something, that means you are making it. That's why we're talking about protein synthesis. All of these terms that you see here are correlated to that of protein synthesis. Okay? Now, is there a large amount of genes or proteins in our body, or not quite so many? There's actually a lot. So to make sense of this, how many letters are in the English language? 26. Okay? Sometimes I always want to say 25, but no, it's 26. Okay? All the letters A through Z gives you 26. There's lots of words in the English language. Okay? There's 20 different amino acids that code for everything that belongs that makes a Brandon, or a Brandon, okay? Or any other aspect. Makes it an Antonio, or Jesse, or any one of you individuals here, okay? DNA is what makes you who you are. Although, if you were cloned with the same type of DNA, are you going to see the same 2 exact people? Probably not. Okay, just because the surroundings can be different. Although it seems like in many identical twins. Maybe they do act similarly. Okay, but anyway, page 336, okay? I don't know why they use the word central dogma. Dogma, I think, just means process. I mean, just maybe a fancy word that they like to use. But anyway, page 336. One of the important features of DNA that remained unresolved beyond the work of Watson and Crick was how DNA served as a genetic code for the synthesis of proteins. Okay, recall that proteins function as structural building blocks for the cells and as enzymes. Geneticists now accept that the basic mechanism of reading and expressing genes is from DNA to RNA to protein. So we see some of these terms up here when we start talking about RNA. These two work together to put this code together. Okay? This chain of events occurs in living things from bacteria to humans. Scientists refer to the mechanisms as the central dogma of biology. DNA codes for RNA, which guides the synthesis of proteins. Okay, RNA, which there are 3 forms. We just haven't really looked at. Oh, yes, we have. We do have one up here. Okay, like I said, there are 3 forms. You have transfer RNA, messenger RNA, and ribosomal RNA. We'll get to that here in just a moment. RNA is a nucleic acid that is similar to DNA. However, RNA contains the sugar ribose. The base uracil replaces thymine and is usually single-stranded. What was the first thing that we said about DNA? Long time ago. Well, I was trying to do this. Okay, remember, DNA is double-stranded. What is the command center of every cell in your body? What directs every cellular activity? The nucleus does that. So it's this molecule that's double-stranded, that's inside the nucleus, that tells that cell what to do. Okay? Single-stranded. 3 major types of RNA are found in living cells. Messenger RNA, or mRNA. Molecules are long strands of RNA nucleotides that are formed complementary to one strand of DNA. Okay? The process is very, very similar. What does it mean to be complementary, do you think? You have to know where you're at to know if close, coming or going. So what that means when they're complementary, again, you have to know where you're at. Adenine bonds to thymine. To know if you're coming or going. So cytosine bonds to guanine. Okay. So, form complementary to one strand of DNA. They travel from the nucleus. DNA cannot leave the nucleus. That's just telling the cell what to do. That's where the directions are. To the ribosome to direct the synthesis of specific protein. Ribosomal RNA, or rRNA, is a type of RNA that associates with protein to form ribosomes in the cytoplasm. The third type of RNA, transfer RNA, are smaller segments of RNA nucleotides that transfer amino acids to the ribosome. Table 2 compares the structures and functions to the 3 types of RNA. The first process, transcriptions. Okay? If we were to look at, now I know we're crossing disciplines here, but back in medieval times, okay, who was a scribe? Does anyone know who a scribe might have been? What does a scribe do?
They just pretty much write down everything.
Okay, they can read and write. Could everyone read and write in the world back in, whether it's Roman times up to maybe 1000 AD? No. So you would have probably past 1000 AD. Middle Ages probably go to what, maybe 1200, maybe? I don't know. You have to get past the Renaissance and then, okay? Are you done?
Yep, sorry.
Oh, okay. All right. So you were right. A scribe is someone who could read or write, okay? So what molecule, what do you suppose RNA is going to do? If it's reading the directions, Again, what is directing all cellular activities? What molecule? It's in the nucleus. Did I hit it? Am I close?
Yeah.
I just know I had to reach up high.
Just a little bit down.
A little bit down. Go a little lower.
How-
Nope.
There we go. Actually, I cheated. I was looking at the camera. I was just guiding myself that way. So again, DNA cannot leave the nucleus. So that directions from the nucleus has to get to the ribosome somehow. So the message has to get there. That's what messenger RNA does. So very briefly, we're going to take this in stages. Okay. All right. Get your drink of water. All right. So transcription. The first step of the central dogma involves the synthesis of messenger RNA from DNA in a process called transcription. So we get the term scribe or transcription meaning to read. Okay? So a little bit of a review. Someone give me some nucleotides. Okay, let's see here. Okay, 2 more. Okay. Now, it was a very subtle hint. Okay. Could this be half of a molecule of RNA? The answer is no. Okay, because it's got thymine in it. Okay. So now our complementary base pairs of DNA, okay, so what are our complementary base pairs? Adenine is, and again, you are responsible for this, C, T, okay, so now, we know that this is DNA because this is double-stranded. Okay? Reason we say double-stranded because this is a strand on top, this is a strand on the bottom. Okay? Do we happen to remember, I guess we drew it like this with dots. Does anyone happen to know what these dots might represent? It's a type of bonding. We've only said it once or twice. The simplest element in the universe. Hydrogen. This is hydrogen bonding that holds these together. Okay? Now, if we were to replicate it, if you were to replicate DNA, there is a specific enzyme. We put a circle and we had it going in that direction. It was DNA something. What was that called? DNA helicase. And if this is sounding not familiar, you probably have this wrote down somewhere on your paper. Would that be correct to presume that? Who does have that wrote on their paper? I'd like to think that everyone would have their hands up. And if we're going like this or shaking our head no, I'm kind of perplexed by that on why we wouldn't write that down, but that is your choice. But here in transcription, okay, let's see here. Okay, when you look up at figure 13, okay, notice you have a molecule of DNA there because you have a top and a bottom, correct, on figure 13. Okay, now, what is that green bubble or green circle? Okay, how do you know that that's an enzyme? Other than we just told you that it is. Say that, one of you two said that. The N in ASE. That means it's an enzyme. So in this case, you have not DNA helicase, but rather RNA polymerase. Is that one of your vocab terms? It is. Okay? So this RNA polymerase is also an enzyme that's going to break this apart. Okay? All I'm going to do, this was just for added practice. I'm going to break this apart and I'm going to slide it down like what you see in figure 13. Is anybody lost right now? So this is all that we're doing. This is just going to go down a little lower, and I should have had more space in here. That's my mistake, your fault.
Your mistake, your fault.
No, my mistake, your fault. Well, I didn't do anything wrong. You don't know what I'm thinking?
Oh my God.
You don't know what I'm... Okay, that's probably a good idea. Okay. So again, all we're doing, we're just moving this down just a little bit. Because what do you notice? What is something significant about messenger RNA? Is that double-stranded according to this diagram on the top of page 337? Does that have a top and a bottom? No, RNA is only single-stranded. Okay? So what that means is we're sliding this down so that we can actually start reading this code. Because over here, what did you say? What does a scribe do? Reads and writes. So that's in this process we call transcription. So the bases are also complementary, except there is no thymine. It's uracil instead. So we can still see adenine, we just don't see any thymine. So the complementary base pairings would be the same. So this would still be guanine, this would still be cytosine, but this is no longer thymine, it's something else. U. It's U. It's U. It's U. What's the abbreviation for uracil? Me? Oh, the letter U. Oh, okay. So notice you can still have adenine. I missed one. Which one are we talking about here? This one. Oh, okay. I thought you were talking about a specific letter. Oh. Okay. Oops, oh, yeah. Okay. There we go. Now we got it right. I was just seeing if anybody's paying attention. Actually, no. I made a mistake. Okay? Yeah. See, this is where you say, your mistake, my fault. And that's fine. I'm okay with it. I'm the one that messed that up. Okay? So what that means is this is going to be a specific meaning. Did you notice every time we did that DNA replication, how many letters long was it? If you don't know, count them on what you wrote down last Thursday and Friday. How many?
12.
Okay. 12. We've done it in 9. We've done it in 12. Both of them have a factor in common. The number 3. because codons and anticodons come in letters of 3. And that's why we got up to 7. I said, okay, I want 2 more to get to 9, which is also a factor of 3. Because tomorrow, what we're going to do is we're trying to put this process together. We're going to talk about it lecturing. I thought it was worth our time to read about it, see it, and then when we talk about it, hopefully that reinforces that. Turn the page to 338, okay? What do you see there? It's a table, or figure 14. Those are all of the different amino acids that get coded for that make proteins and also enzymes and everything else in DNA. It's one of the things you will be responsible for interpreting. It doesn't mean anything to you right now. The reason that it doesn't is because you haven't learned it yet. You just don't know this material yet because you haven't been taught that. Okay? That's where we're going to put these 3-letter base pairs to use. Okay, so we'll start with that tomorrow. Okay, keep working on those vocab terms. So we got 1, 2, 3, 4, 5 done. All right, we'll catch up to you next time.